Very good morning, everyone. Thank you for joining our webinar, Revealing the Complexity of RNA Biology, Solutions for Discovery and Therapeutic Innovations. Again, this is Irene. I'll be your host for today. Without further ado, I would like to introduce the speakers. So far, we have two speakers for today. And firstly, I'd like to introduce Wayne Doyle, Head of Scientific Platforms and Strategy from Eclipsebio. He will be sharing how Eclipsebio portfolio of sequencing-based assays and integrated workflows reveal the true complexity of RNA biology and transform them into actionable insights. So aside of Wayne, we also have a second speaker, Ankita, our account manager from Singleron, where she will be sharing some insights on how to optimize your single-cell analysis workflow. If you have any questions, you can always use the Q&A function at the bottom of your Zoom window, and we will address all Q&A questions after both of our speakers finish their presentation. And without further ado, let's invite Wayne. Wayne, you can share your slides, please.
Thank you very much for that introduction.
No problem.
I'm excited to talk to you all today about how Eclipsebio provides support for looking at different aspects of RNA biology across multiple different dimensions using sequencing technologies. So I want to start by just giving an introduction to who we are at Eclipsebio. So we were founded in 2017, and over the past almost 9 years, we have been working with different partners, both academics and drug developers, to help reveal different aspects of RNA biology, and then also support designing therapies to help fix issues with RNA biology diseases, and then also validate those drugs very comprehensively. So in here are just some examples on therapeutic space, but we also provide a lot of support for academic research to understand what's the dynamics of RNA binding proteins. How does RNA structure change between different conditions? And so for today, I'm just going to show a few examples of that support. Starting first by just giving kind of an overview of RNA. So although we often think of RNA as just a single sequence encoding a protein, RNA truly is a very dynamic molecule with a number of different aspects to its biology. This includes secondary structure on how an RNA actually folds in the actual cell environment that can affect the rate of translation, its overall stability, its regulation by other factors, as well as protein translation. That's a very dynamic process. Ribosomes speed up, slow down, and that can change from cell type to cell type across disease conditions. And then also regulation, how specific proteins are binding, where different methylation marks are deposited, and where microRNAs can bind and do regulation. And then a final component, also looking at what UTRs are specifically expressed and used. We have observed, and others have observed, that between different disease conditions, such as cancer and healthy, you can get very large changes in the UTR landscape. And so at Eclipsebio, we provide analytical support to really look at all of these dimensions of RNA biology. But today I'm going to focus specifically on a few vignettes looking at different components, starting first with looking at secondary structure of RNA. So all RNAs do fold into very complex secondary and tertiary structures, and these structures can play key regulation in multiple aspects of biology. Today I'm going to focus a little bit on translation first. And so this is work that came out last year in which researchers were looking at why some RNAs that are expressed in cancer are able to maintain translation even in a hypoxic environment. Most RNAs under hypoxia will have decreased translation as the cell compensates, but some cancer-related genes maintain that translation. And what these researchers found is that's actually due to the formation of a specific secondary structure, which we can see here, that enable translation to occur even in a tumor environment. And so that can help understand kind of the biology of the cancer system itself. But secondary structure also plays key roles in therapeutic development. This is from research now actually over 20 years ago, in which they looked to see with siRNAs, how does the actual secondary structure affect the ability of an siRNA to do knockdown? And so moving along the right axis, the x-axis on the bottom here, more to the right indicates that an RNA region has more accessibility. In other words, there's more unpaired bases that are present. And the more unpaired bases that are present, the more efficacious an siRNA is in actually decreasing the amount of protein expression. If you move more to the left and there's more of kind of a rod, that there's a lot of base pairing occurring, you have a lot less knockdown. And so due to this, we have supported a lot of partners that are looking to see what's the actual secondary structure that my RNA of interest is having in my specific model system. And typically our partners come to us because they first want to look at structure using algorithmic approach. And this is a very common approach in which you take the sequence and then you predict what's the actual structure based off that sequence. But the problem is that that's not necessarily what the actual structure is going to be in the cell if you use an algorithm alone. And the reason for that is that the actual structure that an RNA folds into depends on a number of different, very cell-specific effects. It depends on how quickly is the RNA actually being produced in the nucleus. Are any modifications added to it? Is it put into a cellular compartment with specific ion levels or specific lipid concentration? And then even within the cytoplasm, the rate of translation, binding by cellular factors such as proteins, can all change that secondary structure. And so this then makes algorithmic approaches very difficult because then you're not able to actually include this information. Most approaches can't predict this protein bound, so it's going to change in this approach. And so what we do is we actually do a direct measurement of the RNA secondary structure, actually within the context of the cell, to see how it's behaving. And this approach is an optimized shape map approach in which we can take an RNA really in any context. It can be in buffer solutions, so just seeing how the RNA naturally folds. If it's a IVT RNA, such as a vaccine, we can also look at the structure within a lipid nanoparticle for delivery, or we can look at it within the actual cellular context itself. And so we keep that RNA in the specific context we want to examine, and then we treat with a chemical reagent that can interact with any base that's unpaired. So anything that's accessible, for example, within a loop, that chemical can come in, add a small chemical adduct to those bases. Anything that's paired, for example, within a helix, that chemical can't interact, and that prevents a chemical adduct from being added. We then extract the RNA and perform cDNA, and then we do next-generation sequencing. And anywhere that that chemical reagent interacted, so anywhere that's an unpaired base that's open in the secondary structure, that then appears as a mutation. And so you can see that here, we're moving on the x-axis is each nucleotide position along an RNA. We have two controls just to account for any potential sequencing errors, or if this is an IVT RNA, account for fidelity issues. But of interest here is the reagent treated. And so anywhere that we see an increasing teal color, that indicates an increased mutation rate. And so that's the site where this chemical reagent was able to come in, interact with the base that was open. And so this is, for example, a region that's unpaired and consistent with a large loop. And then where we get drops of color, those are regions that are more consistent with the helix, a region that had a lot of base pairing. And so from this, we can go across the RNA and say, this base is paired, this base is unpaired, and then use that to determine what's the actual secondary structure of the RNA in a specific environment. And so this is the same RNA I shown before as an example. We're zooming in. On the left is the RNA. If we just take the sequence alone, give it to a computational algorithm and say, what would the expected structure of this be? And I'm highlighting one small subregion near the top. And then on the right is the actual structure based off probing within the cell itself. So this is in the context of proteins that are present, the cellular environment, microRNAs binding. And as you can see, there's a very large structural rearrangement that occurred here. Structural algorithms tend to put bases that are close in order. So base 1100 is then close to 1140 and so on. But actually within the cell itself, you can get these large rearrangements that the base 600 is very close actually to base 1400 in actual two-dimensional and three-dimensional space. This can have large impacts on the actual translation of the RNA, the ability of different proteins to bind, and really affect how this RNA actually behaves. And so this is an approach that we use very frequently for our partners that are looking to understand the actual biology of a specific gene, but also in a therapeutic space to understand what is an ideal region to target, what's actually unpaired within the system itself. But for today, I was going to show a more concrete example of how looking at comparisons in structure between two different conditions can help reveal a key aspect of RNA biology. And for that, we're going to do a comparison of looking at RNA secondary structure within a buffer condition. So this is looking at how the RNA just naturally folds. Nothing's present. It's just the base pairing that's going to be most cohesive under thermodynamic principles to create a structure. We also then looked at the same RNA actually within the cellular environment. So this is in the context of proteins being present, microRNAs binding. And then we looked to compare, are there structural differences and changes in base pairing that occur between the two? This then can allow, if we look at individual bases, actually see protein binding effects actually occurring on base accessibility. And so I'm going to show an example of a gene called ferritin. And this is an RNA that is bound by RRP1. It's an RNA binding protein. And it binds at two specific sites. It binds near the top of the RNA at a G16 and A15. And then it also binds at this position C8. And so those two loops are key based off X-ray crystallography, known sites where this RNA binding protein can bind. Using our E-shape technology, we're also able to visualize this. Where I'm showing here, each circle is an individual base. And then we're showing the structure as predicted based off within the cellular environment itself. But we've now colored by something called delta shape. And delta shape is where we take the amount that a base is able to interact with that chemical reagent that only interacts with unpaired bases. So we look at how much does it react within the cell and how much does it react outside of the cell. If a protein's binding in a specific site, that occludes that chemical from coming in. And so that will appear as an increase in this delta shape reactivity. And so we look across those two unpaired loops that are known to bind by RRP1, both have high delta shape. So they're both very blocked within the cell, indicating that RRP1 has actually bound to these two sites and is having its activity, all without doing really any type of pull down and allowing us, we can then look across the entire transcriptome and see these pinpoint changes as well, allowing really an unbiased screen of what proteins are binding across everything. And we can pair this also with motif screening and say we've seen a change. Is this something that's associated with a known RNA binding protein that could be acting in a specific system? This is something that we do frequently as kind of a first pass screen, where we'll say here are indications that an RNA is being regulated in a specific system. But then to do validation, we compare with another one of our assays, which is called RBP Eclip. It's part of our Eclip family of assays, in which if we suspect that an RNA binding protein is binding to a specific site, or if we're just interested in seeing everywhere that a given RNA binding protein binds, we can use an antibody that's selective for that RNA binding protein. We then cross-link the RNA to all the proteins that are present, use that antibody to then enrich for that protein and its bound RNA. We then generate a sequencing library where we enrich specifically for those RNAs that are bound by that protein of interest. And then we do a variety of bioinformatics analyses to see where and how are those proteins binding. And so this can be used as a validation of what we saw with that E-shape assay of looking at the structure. I'm showing here actual output of the RBP Eclip assay. This is a browser track, so it's showing the actual sequencing reads that are obtained, specifically from 4 different RNA binding proteins. On the bottom is a gene model. So this is looking at the gene WSB1, where each of the thick squares is a different exon. The thin lines are then introns, and then these are the potential isoforms that have been annotated in the genome. This is then an input, so this is just a control to see if we're getting non-specific enrichment. Our V-clip is a very clean assay, so we're not seeing much of all noise being present. And then we have the 4 RNA binding proteins that we examined, LIN28B, RBFOX2, SF30B4, and ZC3H11A. Each of those binds to a distinct site. What we're seeing here are what are called read polyps. So these are the regions of the RNA that are directly associated with that RNA binding protein. And so you can see, for example, that LIN28B is an intronic binder. It's binding specifically at this site, but that's distinct from RBFOX2, which is also distinct from the final RNA binding protein as well. And so we can then use this to look across and say, what are the actual genes that are being regulated by a given RNA binding protein? Or how does that change if a disease state is present, or if you give a chemical drug, can you see changes in those RNA binding protein patterns? We also, using our MirrorClip Plus assay, we have an optimized version of this that can actually look at microRNA binding itself, in which we enrich for the risk complex. and then do a library preparation in which we can directly look at the microRNA reads that are within that RISC complex and their target mRNAs. And so the output of that is shown here, where each of these browser tracks is a distinct microRNA that we're able to detect binding to the 3' UTR of E2F3. If this is direct measurement, we're actually looking at the reads, identifying that they are microRNAs. This isn't a computational prediction. And then we're able to see that each microRNA binds to a distinct site. And the relative height also tells us the relative amount of that microRNA that's bound to this gene, allowing us to then compare across and see microRNA regulatory networks that are present. Since this is an assay that enriches for the RISC complex, we also can use this to look at sRNA off-targeting, which we enrich and see where does every sRNA bind, allowing for a real strong profile of off-target activity across the system. And so that was some examples of looking at both RNA secondary structure and then using that to inform a potential indication that an RNA binding protein is present, and then using our Eclip assays to profile it. But that's just one small slice of our capabilities to look at RNA biology. The final story that I was going to talk about is actually looking at translation, which can be impacted by the secondary structure, but also impacted by specific RNA binding proteins as well. And when we think about protein translation, often our partners are focused either on the level of the RNA itself or the level of the protein. It's more challenging to actually look at the dynamics of translation itself. But importantly, therapeutics and diseases don't only affect level expression, they can have very fine-tuned effects on translation itself, leading to ribosome pauses, for example. And so we found it can be extremely beneficial to actually look at where are ribosomes speeding up, where are they slowing down. And to do that, we use an optimized version of ribosome profiling, also called RiboSeq, in which we take a sample. There's going to be some RNAs that are being translated, some RNAs that aren't. And then we split that sample in half. For half of it, we extract out all the RNA, and then we do a typical RNA sequencing library. And so this tells us then the relative amount of all the RNA that's present. Is a given RNA expressed more than a housekeeping gene like GAPDH, for example? Is expressed a lot less? But importantly, for the other half of the sample, we lock the ribosomes in place, and then we digest all the free RNA. So the only RNA left is directly within the ribosome complex. Looking at the number of sequencing reads that we obtain here then tells us the relative amount of ribosome association. So is a given gene being very well translated? Is it being very low translated? But also, since we're looking at the RNA directly within the ribosome complex, this also gives us nucleotide level positioning to say this is exactly where ribosomes tend to pause or where they tend to speed up. Which then after sequencing, we can do a variety of analyses. I'm showing on the right here, this is analysis just looking at bulk changes in translation, where you're treated with a drug that blocks translation, and that's shown here on the volcano plot. Each dot is an individual gene. And then the left is changes in RNA-seq, so the changes in the actual gene expression. This is a translation-specific drug, so we see no changes in the actual expression. We do see strong suppression as shown by the colored blue dots when this drug is present at a significant level. But I'm going to focus more on a refined analysis where we actually look at where ribosomes pause. And this is based off an analysis of a drug that Pfizer developed in 2017 called PF846. And this was a drug developed to treat cholesterol disease. It's a small molecule that is able to enter the exit channel of a ribosome. For the vast majority of RNAs that are being translated, this drug has no effect. But in a very small subset of proteins that are being generated, this small molecule interacts with the nascent chain that is being generated. It then leads to a conformational change, and then the ribosome gets stuck. The ribosome can't move forward. The nascent chain can't exit. And so that then leads to a ribosome stall, preventing protein production. This is a very specific effect. It only affects 22 genes. The target is PCSK9. And so we then asked, can we actually see these ribosome pauses occurring at the expected site using this assay? And so I'm showing the results of that experiment here, where again, this is a browser track. At the bottom is the gene PCSK9. The thin lines, again, are introns. The rectangles are thick rectangles and squares are exons. And the thinner rectangles at the end are the UTRs. And I'm first showing the data for ribosome-protected fragments, so the RNAs that are directly within the ribosome complex. for a control in which we see that there's data on every single exon, indicating very robust translation. Ribosomes can move along very quickly, generating protein production. If we look when the drug is introduced, we see a strong enrichment right on the first exon. This is where this drug is expected to act. And we see very little translation occurring at the subsequent exons. And importantly, we see no effect on the actual RNA-seq. So the transcription is the same, the gene expression is the same. We have equal coverage from UTR to UTR and expression on every single exon. So this is then providing a very specific effect to just act on translation just at one specific site. And we have developed an algorithmic approach that allows us to screen across the entire translatome and identify these stall sites statistically. So even if you don't expect that your drug could be affecting stalling, our analysis can capture that to give a wider view of what's actually occurring. We also can do that between different disease conditions as well to see how stalling patterns change. And so that was a quick overview of some of our capabilities to support looking at different aspects of RNA biology. But as I mentioned at the beginning, RNA is multidimensional, structure matters, translation, microRNA binding. And we have developed a suite of assays that profile each of these different aspects. Our E-shape assay to look at secondary structure, which I showed today, as well as our eRiboPro assay, our optimized RiboSeq to look at translation, RBP-Eclip and MIR-Eclip to look at protein binding and microRNA binding. For the sake of time, I didn't go through it today, but we also have assays to look at methylation, either at nucleotide level resolution or MIRIP optimized assays to look at regions that are methylated as well. And then our NSeq assay can be used to identify what specific UTRs are active in a specific disease state as well, both 5' and 3' UTRs. And for all of these assays, we provide this through our lab services team, in which we provide support to measure different aspects of RNA biology, so to really understand how does the system work, what's actually occurring in it. But then we also provide support for therapeutic development for both target discovery and validation of small molecule and small oligo therapeutics. Outside this, we also do support IVT RNAs through our eMERGE platform, in which we can look to see how is the IVT RNA behaving, are there optimizations that can be done, or is it good to move into the clinic? But focusing specifically on our lab services, the typical workflow that we have is that you will meet with our support team, review what your goals are, which of our assays we can apply to it to make sure that you achieve what you need to. You then submit your samples to our team. They then perform all the wet lab processing for you, do stringent quality control, and then our bioinformatics team will analyze your data. You'll, at the end, receive your raw data, but also secondary analyses that we've curated, as well as interactive reports that I'm showing a screenshot of here, and that you can go through the data, actually explore it, see what results were obtained. With that, I'm happy to end and transfer over to Ankita.
Thanks, Wayne. Let me just share the screen real quick. Okay. With that to say, thanks, Wayne. Sorry. Thanks, Wayne, for the informative presentation. And again, for those who have questions, you can drop your questions by clicking on the Q&A icon at the bottom of your Zoom window. And as mentioned, let's invite our second speaker, Ankita, to talk about single-cell workflow. Ankita?
Hi, everybody. Good morning. Thanks a lot for taking the time to join this webinar. Just a quick introduction about Singleron. We're a single-cell solutions company with a global presence, and Singapore is our regional APAC headquarters. In terms of our solutions, we provide end-to-end solutions for our single-cell workflow, starting with tissue preservation, dissociation, single-cell capture, partitioning, the mRNA capture, library prep, sequencing, and bioinformatics. And we have a lab present in Singapore as well. So it's not just kits and instruments, but you can talk to us about your samples, and we can take it forward from there, providing you a complete one-stop shop solution for your single cell projects. All right.
Thank you, Ankita, for the presentation. It's
Thank
very
you.
short and sweet, but it's very informative. And again, if you have any questions, please drop them by clicking on the Q&A icon at the bottom of your Zoom window. Just to get the conversation started, we would like to just check in with Wayne on just a curious question. Is ribosome pausing the most common type of effect you guys observe?
Yeah, so I'd say it's a mixture between looking at just general ribosome association, that's also called translation efficiency in the literature. I'd say probably the little bit of majority is more focused on that, just seeing if you give a driven drug, does that change kind of the overall rate of translation? And the ribosome pausing typically tends to be a little bit more refined analysis, although we do see that very frequently in the IVT RNA space to look to see codon optimization. Is there a region that is potentially problematic, leading to a pause that could be optimized?
Ah, I see. Okay, thanks. There's one question coming from the attendees. This is for Ankita. Just curious, what would be the sequencing depth requirements for single cell sequencing from Singleron?
Usually we aim to capture about 20,000 cells per sample with our platform. And so we recommend about 35,000 reads per cell. But of course, this is dependent on your sample type and your research questions, so we can take it on a case-to-case basis as well.
Okay, sure. I think at this point, there's no further questions. And hence, I think it's safe to say that we are getting into the end of the webinar. So if you have any more questions on single cell sequencing or protein RNA, do contact us at sales.sg@Singleron.bio. You can also get the email by copying from the chat, or after this webinar, we will actually send up a follow-up email with the link to the webinar so that you guys can watch it on demand. Follow us on LinkedIn and X to stay tuned. Thank you everyone for joining us today, and see you guys in our next event. Thank you very much.
Thank you.
Thank you.